Today the topic under discussion is gene interactions. This is for class 12. Now, as you know, that gene interactions are the interaction that takes place in between two allelic genes or in between two non-allelic genes. So we express gene interaction in form of intragenic gene interaction, which is in between an allelic gene. Say for example, which generally we say in monohybrid cross, we have one allelic pair, and this allelic pair are expressed in form of dominant and recessive. In intergenic gene interaction, it's a non-allelic gene. That means here the genes will be two different characters. Now this is generally found among the dihybrid cross, where two different non-allelic genes interfere among themselves and give certain characters which are not in accordance with the Mendel's law. Now, in intragenic gene interactions, what are the examples? Incomplete dominance, codominance, multiple alleles, and pleiotropy. In intergenic or non-allelic gene interactions, epistasis in different form, polygenic traits, complementary genes. Now, first we will go in one of the intragenic gene interaction, what we call incomplete dominance. In incomplete dominance, we see in some examples are like, for example, 4 o'clock plant, which is known as Mirabilis jalapa. 4 o'clock plant, we have two varieties. One is red and the other is white. Red is dominant, so depicted by capital R in a pure line. White is smaller, and when these two form the gametes, they give capital R, and here it is smaller, which fertilizes to produce capital R and smaller. Now, if they would follow the Mendel's law, according to the law of dominance, it should be red, but this does not happen. This capital R and small r appears to be pink, which is an intermediate expression. Now, if we again self-cross it, self-crossing means crossing with the similar genotype. In the F2 generations, we have capital R1, capital R 2, and small r 1. That means it comes in the ratio of 1:2:1. 1 red, 2 pink, and 1 white. And the genotypic also same ratio, same capital R is to capital R is to small r is 1:2:1. Now, this is an example, a classical example of incomplete dominance. You find this also in Snapdragon, which is called antirrhinum. We take an example of the incomplete dominance also in a fowl called Andalusian fowl, where the male is found in form of white splashed, expressed by capital W, black, small w. Now, when they are crossed in the F1 generation, you have capital W, small w, which should be expressed, if they follow the Mendel's law, should be white, but it doesn't appear white. It is a slate color, which is different. It's neither the male or the female. It's a different color. When you self-cross it, again you find the white is to slate is to black is 1:2:1. Now, this is also an example of incomplete dominance. We go to the second example of the intragenic gene interaction is codominance. Example is found in certain cattle. You will see that in cattle, some cattle have black coat, some cattle have white coat. In black, expressed by capital B as a dominant, and in white small b as a recessive. Now when they together form the gamete, capital B comes from here, small b comes in the gamete. When they fertilize, it forms capital B, small b, which is a rowan in color. That means it's expressed as it gives both the it is expressed in both forms. Now, example also we find in the blood group in human being. We have 3 genes, IA, IB and I, and they are expressed in 6 allelic forms, which are the 6 multiple alleles like IA, small I. This is a dominant gene. IB, both dominant, IB, small I, one dominant, one recessive, and IB and small I, which is a recessive gene, which is expressed generally in 4 forms, like we say A blood group, B blood group, AB blood group and O blood group. But codominance is seen when we have 2 genes like IA and IB existing together. They are expressed as AB. So that means here A and B both are expressive. That means here when we find in the genotype, they are expressed as the AB blood group, where IA gene, A and IB gene, which contribute to both these characters, are existing together. So that type of factor is called a codominance. Now, there are certain other things like, for example, pleiotropy. Pleiotropy is the ability of the gene to influence more than one trait. That means, like say for example, phenylketonuria. Phenylketonuria, this is a gene defect which is caused in the 12th chromosome, which does not form a particular enzyme that converts phenylalanine to tyrosine. Now, due to absence of that gene, the phenylalanine accumulates in the body of the individual and it causes certain characters like mental retardation, the muscle breakdown, and that means there are several factors which are expressed due to one defect, one particular defect of the gene causing several types of defects or several expressed in different ways. Same is in case of sickle cell anemia. This is also a defect in the hemoglobin molecule, where the hemoglobin molecule is not formed. So people having sickle cell anemia, they suffer from anemia. Oxygen cannot be carried properly in these people. At the same time, sickle cell anemic persons are resistant to malaria because the malarial germ or the parasite Plasmodium cannot complete its life cycle in the RBC because of the absence of hemoglobin. So we see several characteristics is expressed in several forms. One particular defect of the gene, so it can influence more than one trait. So these are the things which we study today. Let us see the assignments. Please do the assignment. Please write it down. We are going to see later on. We'll check it out. Like A, define number 1 pleiotropy, number 2 codominance. So pleiotropy and codominance. I am repeating. B, what are the intergenic gene expressions? C, explain incomplete dominance with examples from of Andalusian fowl. Thank you very much. Have a nice day and God bless you.